Hey, welcome to this video. My name is Callie Chappelle, and this video is about bacterial genetics. It's made for Bio 305, which is Introduction to Genetics at the University of Michigan. So let's get started. In this video, here's a quick and dirty. We're going to go over bacterial recombination, plasmids, the F+ strain, the Hfr strain, minute mapping, and finally the F' strain. So what is bacterial recombination? Well, this is not the first video that I made about bacteria, so if you needed any review, go back and rewatch the last one. But there are two big types. The first is integration of linear DNA, which you can see here, and then the integration of circular DNA or plasmid. So let's talk about integration of linear DNA first. Well, first, if you want to put this piece of integrated DNA or DNA that we want to recombine into the bacterial chromosome, which is this green one, it's going to require two crossing over events. Okay, so let's draw that right here. Yeah, let's do this. It requires two crossing over events, one right here and one right here. And it's not expressed unless it's integrated into the bacterial chromosome. So if you just have, let's see if I can go back. I guess I can't, never mind. If you just have this chromosome or like this piece of DNA just hanging out inside of the bacteria, it's not going, any genes that are on this expressed, so for example, if there's like the A gene or something, it's not going to be expressed. However, if you integrate circular DNA, or what we would call plasmid, into a bacterial cell, that first only requires one crossing over event, so it's one crossing over event like this, and it can be expressed even if it's not integrated into the bacterial chromosome. So if plasmid has the A gene, then the A gene in this cell will be expressed, even though it is not integrated into this, which is the bacterial chromosome. Alright, so what are plasmids? Well, plasmid is just a circular DNA that's carried and transmitted among bacterial cells. So it's kind of like a genetic satellite. So you can see if this is a bacterial DNA, which we know is double-stranded and circular, plasmids are also circular. They're generally much smaller, and they're also double-stranded. Now, they are not considered part of the bacterial chromosome or genome, which you can see it's not in the bacterial DNA, although it can affect the phenotype of the bacteria by coding for proteins, etc. In fact, this is something that you should definitely know. If the bacterial chromosome is mutated for some reason, so let's say that this is supposed to produce, I don't know, we have a gene here that, I don't know, does not provide antibiotic resistance, but this plasmid has a gene that provides antibiotic resistance, then this entire cell will be antibiotic resistant. Okay, so if the bacterial chromosome is mutated, it can't produce something, then you could actually add a plasmid that produces it, and the bacteria will appear wild type. And then there are two different types of plasmids. There's single copy, where you have just one plasmid in the cell. So like this cell would look like this, where here's the bacterial genome, and here's just one plasmid. Or multi-copy, where you have many plasmids. So if this is a bacteria, and there's the bacterial chromosome, we have many plasmids. Alright, and then you also have conjugated, which means that they can initiate conjugation. So an example of this is the F plasmid or the F+ plasmid. Or non-conjugated, which means that they can't initiate conjugation. So that's just a plasmid that's hanging out and doesn't create a bacteria, it doesn't make a bacteria create a pili, that's a pili. I think there's two L's, but don't quote me on that. Okay, so F+ strains. So there are three different strains that we're going to talk about: the F+ strain, the Hfr strain, and then the F' strain. So F+ strains are bacteria that have the fertility factor or the F+ plasmid. So what is the F+ plasmid? That's what this is a picture of. Well, it's a plasmid that's frequently transferred between bacteria. So it rarely contains genomic material, actually, so it's not going to contain genes that code for various proteins or whatever, that contain genes that code for various proteins, but it is going to be something that controls whether or not a bacteria is considered F+ or F-. So that is what the F+ plasmid does in terms if you are considered an F+ or an F-. Now, I think this is kind of a bad metaphor, but you can think about it if you're a boy or a girl. It's controlled by whether or not you have a plasmid. Obviously, bacteria don't have gender, so it's kind of a stilted analogy, but you can think about there being two types of bacteria, those with F+ plasmids and those without, or considered F-, right? So bacteria with the F+ plasmid will replicate and donate F+ plasmid to a bacteria without one. So in this case, if you have a bacteria with an F+ plasmid, and then you have a bacteria without one, then the F+ plasmid in the one with an F+ plasmid will replicate and get transferred. So here's a picture of this, right? So you have a cell, so we got two separate bacteria. This one has an F+ this one has an F plasmid and this one does not, then the F plasmid gets replicated. It in conjugation would have a mating bridge. The F plasmid gets transferred. So here, remember, here's our F plus cell. Here is the plasmid getting transferred, the F plasmid getting transferred, and then finally, after conjugation happens, then we're left with two F plus cells. Okay, so an F plus crossed with an F minus results in an F plus, right? So here's we have an F plus, here's an F minus. It results in two F pluses. If you just have an F minus and F minus, obviously you're gonna end up with an F minus because we're not transferring anything because neither of these have the F plasmid. And then if you have an F plus and you have another cell with an F plus, then you just result with both having F pluses, but there's no transfer, obviously because both of them already have the F plasmid. So the F plasmid is only transferred from a bacteria with an F plasmid to a bacteria without an F plasmid, so the final result is both have F plasmids. Alright, so we also have this thing called an Hfr strain. So an Hfr strain, or a high frequency of recombination strain, are bacteria that will transfer their entire genome. So where does that come from? Well, it occurs when an F plasmid, it gets integrated into the bacterial genome. So that's like when this happens here. So you have a bacterial genome, you have an F plasmid, and the F plasmid jumps into the bacterial chromosome, okay? So what this does is when you have an F plasmid integrated into a bacterial chromosome, then it allows the bacteria to replicate and then transfer its genome to another bacteria, okay? So how does this work? Well, there are three big steps. So please ignore my really terrible diagram here because I'm not that great at drawing on the computer, and I couldn't find a diagram online that sufficiently showed what I wanted to show here. So this is really what happens from start to finish. The first thing that we do, like I said above, is the integration of the F plasmid into the bacterial genome. So F plasmid with one crossing over event hops into the bacterial genome, resulting in this cell that's ready to go. So this is considered an F plus cell. Alright, so then we have the transfer of all or part of the bacterial chromosome through conjugation. So we have our bacteria that has the F plasmid integrated into the genome. We have replication event. We have this pili forming, P-I-L-L-I forming, and then we have this chromosome starting from the origin of transfer going over here. Oh, that's something that I forgot to say. So we always have an origin of transfer, O-R-I-T, and that's just the point where transfer starts. Okay, so that's transferring over here into this, okay, and then the last step is the integration of that transferred genetic material into a new bacteria. So the new genetic material is double-stranded, and that's just going to get spliced using two crossing over events, or two integration events to get integrated finally into this bacteria. So for example, let's say that the bacterial chromosome, let's say we have the A gene, we just like to say the A gene, or the A plus gene, because that's going to be your grade in genetics, absolutely. So we have the A plus gene, obviously that's not going to change when we integrate the F plasmid, but here, let's say that this is the ORIT. So as this chromosome starts going, right, here, the A gene comes with it, okay? So by the time that, let's say we stop conjugation, and then wow, that was like really loud and obnoxious, and then we have our A plus gene here, then when that A plus gene gets integrated, when this recombinant section of DNA gets integrated into the bacterial chromosome, well, this A plus gene comes with it. So suddenly, you're left with a final cell, a final acceptor cell that has the A plus gene that came from the donor cell originally. So let's compare the F plus strains with Hfr strains. So in F plus strains, they generally do not transfer genetic material, just transfer F plasmid. Whereas the Hfr strain does transfer genetic material. So the F plus strain obviously has the F plus plasmid, where the Hfr strain has the F plasmid integrated into its chromosome. So F plasmid's hopped into the chromosome and is stuck there. So if you have a cross, when you cross an F minus with an F plus, F plus with an F minus, then you're resulting with both F pluses, right? Because here's our So here are two bacteria, one of them which has the F plasmid, and one that does not. Well, when we form a mating fork here, this is going to replicate, and this will go into here. So we're left at the end of the day with, let's try this again. Now we have two bacteria, both of which have F plasmids. And then we're left with two F plus bacteria, where this one was F plus, and this one was F minus. And we're going to compare that to the Hfr strain, where we have an F plus plasmid integrated in the chromosome. So if we have an Hfr times an F minus, you still end up with an F minus. You don't get an F plus at the end of the day, but the entire genome, but now it has the genes of the Hfr. So the final product of this is, so here we're starting out with our same bacteria, and let's say that this bacteria has an A plus, and we're going to cross it with this one. I don't know, let's do this in a different color. Let's cross with this nice green bacteria. Then we'll get a transfer of genetic material, and that'll result in something that looks like this, the integration of some of the genetic material from this orange one, such as the A plus gene, into the genetic bacteria, but there's no change in F plasmid, okay? The entire genome would need to be integrated to make a recipient F plus it would have to pop out to produce a plasmid, pop out of the bacterial genome. Okay, so let's actually compare this with the genes. We already looked at this a little bit, but let's do a really focused analysis, the difference between the F plus strain and the Hfr strain compared with genes. So let's go with the F plus strain first. So we're just starting out with a normal cell that has the F plasmid, the F plus plasmid, and it also has the A gene, which is right here. I'm just going to color that in orange so it's really clear to see. The A plus gene, which is right here, just like we've been doing, okay? So the first thing is the conjugation and transfer of the F factor. So if we cross this, so the cross here would look like F plus A plus cross, so that's this cell, cross F minus A plus, so this is a cell that doesn't have, or F minus A minus, this is the cell that has the A minus allele of the A gene and also does not have an F factor. Then the result is going to be the F plus and the A plus, so this one doesn't change, and then this one, F plus and the A minus, so no genes transferred. And then here with the Hfr strain, the first thing that happens is the insertion of the F factor into the bacterial chromosome. And then we have the same thing, conjugation and chromosome transfer. So we have replication here. This comes over into here. And then we have recombination. We can either have recombination or no recombination when transferred genetic material. When transferred genetic material, such as the A plus gene, is integrated into bacterial chromosome. Sorry, I've abbreviated this a little bit. So what does that look like? Well, let's say now we're going to look at A, B, and C, so three different genes, or three different alleles of the A, B, and C gene respectively, get transferred into this F minus cell like we are looking at right here. Then we can either have two crossing over events where just B and A get integrated, right? So we have recombination, or we don't. We just have two crossing over events here, so in which the product would be this. This would get C plus, B plus, A plus, and then up here we'd have C minus, B minus, A minus, because of there is no recombination here. So in this case, if recombination doesn't happen, then we'll just have be, let's see, F minus, A minus, because the A allele is not getting integrated. But if there is recombination, then the A allele will get integrated, and then this little A will pop out. So the final product will be F minus A plus, F minus A plus, okay? So the big takeaways here are with the F plus strain, there is no genes transferred, so there's no change. The only change is F minus becomes F plus, but there's no change in what the genes are on the chromosome. Now, if you have an Hfr strain, then you do have transfer of genes from the H plus strain to the Hfr strain to the F minus strain. But and on top of genes being transferred, there can either be no recombination, so the genes that get transferred don't actually make it into the bacterial chromosome, or recombination can happen and either all or some of the genes that are transferred from the Hfr strain get integrated into the recipient bacteria. Okay, so the big takeaway here, the questions that you're going to be asked about are minute mapping. So what exactly is minute mapping? Well, when we have our Hfr strain and it's transferring its bacteria into the F minus strain, well, it doesn't always get to transfer its entire genome. Because it gets transferred in a linear fashion, you can actually stop this conjugation at various times and see how many genes have been transferred into this new recipient cell, okay? So when we look at a graph like this shows how many of those genes have been transferred in the order of time. So if this is the origin of transfer, and it's moving like this, well, the genes that are closest to the origin of transfer will get transferred first to the recipient cell, and those genes that are farthest away from the origin of transfer will get transferred last. So if we cut off the time of conjugation really soon, then only the first couple genes will get transferred, and the ones that are lagging really far at the end of the chromosome are not going to get transferred to the recipient cell. However, if you let that go for a long time, then all of them, or many of those genes, even if they're near the end of the chromosome, will get transferred. So when you look at a map like this, the time in minutes goes this way, and then here's the frequency. So really all we care about is when it intersects with the x-axis, okay? Because this is going to show you the initial time in which it's transferred. So we know that if we read it from low time to high time, this is closest to origin of transfer, and this is farthest from origin of transfer, ORIT. Okay, so if I wanted to draw a map of this, if I wanted to write what each of these represents on here, well, the one that's closest to the ORIT is AZI. So that means that this one is AZI because here's the ORIT, and it's coming first into the recipient cell, so that means it's closest to the ORIT. The next one is B, which is TON. Okay, the next one is C, which is LAC, and then the last one is GAL. Okay, so we know the order of the genes that are transferred. So this is really important to understand how to read these graphs. And the big thing is you read it from intersection with the x-axis. You don't actually read it up here. Even though this makes sense, right? Because the y-axis here is frequency of Hfr genetic characters among exoconjugates. So essentially what it's saying is how many of the recipient cells have these integrated. And obviously if this is the first one to go, the most of the cells are going to have this. And then as it goes along, there'll be less and less and less as time goes on. But there will always be the most A because it's the first one that gets transferred, and there will always be the least D because it's the last that gets transferred statistically. That's the reason why there are differing amounts. So how do you do minute mapping? So minute mapping is where we figure out what like we looked at over here, instead of doing it on a line, on a circular plasmid, what the order of the genes are. So what I do is first I always start out writing ORIT and define a direction, okay? So I always like to write it like this, ORIT, and I'm going to say that it's going this way. So what does it mean when I draw a plasmid with ORIT like that? So if this is the ORIT, and that means that when this plasmid replicates, it's going to start leaving from here, okay? So this is the ORIT. So this equals this, okay? So the things that are right here, the things that are directly, which I guess this is poorly drawn actually. I probably shouldn't draw it that way. Just kidding. Hang on, I'm going to erase this. So I realize this is going to be a little bit confusing based on how I drew this picture. Let's draw it like this. There we go. That's a little bit better. ORIT. We're going to draw it like that. So that means the genes that are here, these are going to be the genes that are first to go. And then the gene that's here, so equals first, transferred. And then here is going to be the genes that are last transferred. Last transferred. Okay, so let's figure out what the first and last transferred are. So we know that the gene that's close to the ORIT is going to be the one that comes off first, that's going to be transferred first. So we know that in this case, AZI, or A, is going to be coming first. And it takes 9 minutes, so we're calling this a minute map. So we're going to say the distance between the ORIT and AZI is 9 minutes, okay? And then the distance between the amount of time it takes for A and B to transfer is 1 minute, because 10 minus 9 is 1 minute. So from B to the ORIT is 10. I'm just going to draw this here so you understand, is 10. So that means that the distance between ORIT and TON is 1. Okay, and then the next one is 16. So this is going to be 6, because 6 minus 10 is 16. And here's the LAC. You're a wild type. And the last one is 25. So what is that? That is 14. Yeah, so we have 14. 14 to gal. Okay, so if I wanted to draw this minimap, I would draw it like this. Normally I would, instead of doing the subdivision, I would just write this. So I would write 9, 1, 6, 14. And then sometimes you know the total length and sometimes you don't. So then we just don't know how much time is here. Okay, based on the information that's given. So let's do a sample question with minimapping. So sometimes you're actually not given just one, you're given multiple Hfr strains and you're asked to multiple different genes, and you're asked to draw one whole plasmid. Okay, so how do you do this? Well, you're going to pick a strain to start with. So I'm just going to start with Hfr1 because it's the first thing, and the second thing I'm going to do is draw the ORIT and work around the chromosome. Okay, so I'm going to do the same thing. I'm going to draw my ORIT here, ORIT, origin of transfer here. And then I'm going to start, okay? So first I'm going to look and see what is the very first number. Well, the first number is 10, and wait a minute, F and A, those are at the same exact time. Well, it turns out if they come off at the same exact time, that means they're actually the same gene. So the number of minutes between the ORIT and A/F, because A and F are the same thing, is 10 minutes. Okay, then I'm going to find the next number, so 30. So that means that if I'm going this direction, then 30 is going to be the next number. So 30 minus 10 means the difference, because the distance between B and the ORIT is 30. So the difference between B and A/F is 20. B is here. The next number is 60. So C is going to be here. And then, oops, sorry, this is C. And then 60 minus 20 is what? 20, 30, 40, 50, 60 is 40. Okay, and then D is 70. D, and then the difference between that is 10. Okay, so let's figure out how to do. So cool, we've done this one. We've picked the ORIT and we've worked around the chromosome. Okay, the next is to figure out the direction of the second strain. So the second strain, we're going to start from the lowest number and go to the top number. So that goes B to A to E to C, because we're starting with the lowest number and working our way. So 10 to 30 is A to B, and then 30 to 60 is A to E, and then 60 to 70 is E to D. So we're figuring out the direction of the second strain. It's going to be B to A to E to D. So if I start at B, and I go to A, and then I go to D, and then I go to E, this one's going the same direction. Okay, it's going the same direction. So it takes 10 minutes to get from the ORIT of Hfr, I should write this as number 1, from Hfr2 to B. So that means that if I draw my ORIT for 2 here, ORIT 2, then the difference between here and B is 10. Okay, So what I like to do is I always do this in pencil because I'm going to re-subdivide this. So previously we knew the distance between B and C was 40, but now we have a new thing that's breaking up this 40. So what I want to do is figure out what is the difference between the distance between ORIT and C. Well, I know the distance between B and C is 40, and I know the distance between B and ORIT is 10, so the distance between ORIT and C must be 30. And so I crossed out 40 and I rewrote 30 because you are always going to try to keep these in small increments as possible because it's going to make your life easier in the future, trust me. Okay, so next we're going to draw that strain in and make sure to subdivide section. So I just showed you how to subdivide a section. We're going to do that now. So the next thing, so we're going from B to A, and that's 10 to 30, so 30 minus 10 is 20. Okay, well that's perfect, that's A to F. And then the next thing is A to E. Well, we don't have an E already on here, so we need to put an E in. So I'm going to write E right here. So we know that A to E, I'm just going to draw this in right now, and I'm going to erase it in a second, is 30 minus 60 is 30. Well, we know the distance between A to the ORIT of 1 is 10, so the distance between E to the ORIT of 1 must be 20, because 10 plus 20 is 30, which we already know is the total amount. So I'm going to, just to keep things clean, I'm going to erase this. Okay? And then the next thing that we know, continuing along in our right direction, is from E to D is 60 to 70 is 10, so E to D is 10. So we have our completed minute map, okay? Cool, so the last thing that I want to talk about in this video is the F prime plasmid. So this is not really important, at least in my class we didn't really talk about the F prime plasmid that much, but it was in lecture and it is something you should be familiar with. So sometimes the Hfr strains occur, so Hfr strains we know occur when the F plasmid is integrated into the bacterial chromosome. But sometimes, just like the F plasmid can go into the bacterial chromosome, it can also pop back out. So that's what happens, that's when you have an F prime plasmid. So an F prime plasmid is just when an F plus plasmid pops out of the bacterial chromosome and brings some bacterial genes with it. So here we see that an integrated F, so this is an integrated F plasmid, this pink part, integrated F plasmid, can leave the host chromosome and carry host genes with it. So like, let's say that we have gene A plus here. Well, if this pops out like this, then here's the A plus gene. So the F prime plasmid is the F plus plasmid plus some bacterial genes. Okay? So if we wanted to transfer this F prime plasmid, well, here we have A plus, here we have it replicated and it's moving over here. So at the end of the day, if conjugation happens, then you have this, which is an F plus plasmid with the A plus, and here we have an F minus with let's say this is A minus, with the A minus, then you're left with an F plus plasmid with the A plus, which is this, but also this one, which is F plus A plus as well, because now we have this F plasmid with the A plus gene, and as we know, a plasmid that's integrated can also express a gene that's on the plasmid, okay? So thus genes can also be transferred via the F prime plasmid, not the F plus. So make sure that you understand all of these. Hopefully this video is helpful for you, and hopefully you'll tune into some more. Take care.